can see that here, has grown 2 heads. But the most amazing thing is that if you then again take the middle piece out of that worm and culture it again in plain water with no more manipulation of any kind, and simply let it regenerate, it will once again make a 2-headed animal, despite the fact that it has a normal genome, a normal genomic sequence that we have not edited. And so in perpetuity, as far as we can tell forever, every time you cut these worms, you get a 2-headed animal. So think about what this means. The pattern to which these cells build after damage can be rewritten, can be bioelectrically re-specified towards 2 heads, and in fact now we can set it back to normal as well, in a way that only changes the information stored in this electrical circuit. The DNA is completely normal. We have not changed the genomic sequence of this animal. So we have now found that some information is present in the physiological circuit that is not directly in the genome, and this physiological circuit can store information that is changeable by us. In fact, not only can you ask these worms to make a second head, if you perturb the bioelectric network, you can also get them to build heads that are appropriate to other species of worms. So here you see a normal Dugisia doratocephala with a nice triangular head. By disrupting that bioelectrical gradient, you can make their fragments regenerate flat heads like a Piphalina, or round heads like an S. mediterranea. And not only the external shape of the head changes, but also here in green that you can see the shape of the brain becomes like these other species, and the distribution of their stem cells, which you can see with the red dots, are also changed to be just like these other species. And again, this is done without changing the genomic sequence. So extremely important information is present in this bioelectrical network that guides cell activity towards building specific different kinds of structures.